Hello everyone. Is the sound clear?
Yes.
Okay. Yeah. First of all, thank you very much for having me together with you guys for next half an hour. I'm going to talk about some of our work and how we are trying to understand the situation with ME/CFS, and what I'm going to talk about is mostly based on mitochondrial architecture and how we are trying to use the mitochondrial architecture as a sort of a tool or sort of a biomarker to differentiate ME/CFS from other diseases. So what I'm going to show you basically is two different forms of mitochondria. On your left side is a normal mitochondria, where you can have both network structures of mitochondria as well as a few filamentous mitochondria. But however, on your right side, you can see fragmented mitochondria, which is mostly filamentous mitochondria. And this is the basis of our understanding how ME/CFS is. So as you know that we are trying to find a marker for ME/CFS, and
Bhupesh, sorry to interrupt. Could you possibly share your slides on the screen so that we can see them?
is it not available?
It's not come up. So the green share screen at the bottom.
Next one.
Yes, that's perfect. Thank you.
Oh, sorry. I'm new to this. Okay. So I'll just go back. So on your left side, you can see a normal mitochondria, which is basically a combination of both filamentous mitochondria as well as punctated mitochondria. But on your right side, you can see a completely punctated mitochondria. And this structural difference of mitochondria is what we are trying to see in ME/CFS patients. So as you know that there are a couple of studies done before where the researchers have tried to understand if serum or plasma from ME/CFS patients are taken and healthy cells are allowed to grow in the presence of this serum or plasma, what type of changes happens. And so we tried to do the similar type of experiments. So we took purified serum from patients. We did not take plasma because growing cells in plasma is not that healthy, whereas in presence of serum, we can really grow the cells nicely. So what we see is that a clear difference between the ME/CFS patient serum in comparison to the controlled serums. For example, over here you can see that there are control. This patient is a inherited HH6 person, but healthy. In presence of this serum, there is no change in the mitochondrial architecture. But if you take, for example, here ME/CFS patients, you see that the mitochondria completely changes into a punctated form of mitochondria. And this is what we can quantify. So for example, over here, you can see that if we look into, so we grow cells, the same cells in presence of healthy donor serum, as well as, for here example, 17 serum samples from CFS patients. So you can clearly see that in healthy donors, most of the mitochondria is beyond the average mitochondrial surface area. Whereas in ME/CFS patients, you can see that the mitochondria is mostly fragmented, and the average mitochondrial surface area falls below the size of the mitochondria. So this is a very characteristic feature that we have observed in ME/CFS patients. And not only that, not only the fragmented mitochondria, we also see that the amount of ATP inside the cell also goes down in presence of the ME/CFS patient serum. It's not very significant over here, but still we see that there is a decrease in ATP content in the cell. But there is another interesting feature, which probably ME/CFS patients have realized it long back, that when we take the cells which are grown in the presence of ME/CFS serum and we try to infect these cells with some of the fast-growing viruses, for example, influenza virus or HSP1 virus, we see that these cells are no longer capable of being infected with fast-growing viruses. I am using the term fast-growing viruses here because fast-growing viruses, as soon as they enter into the cell, they need a lot of nutrient from the cell and they need to hijack the cell system to survive. Slow-growing viruses might differ here, but if you look into the data over here, the data is pretty much clear. So, for example, on the upper panel, you see influenza A virus being infected into the cells which are being treated with controlled serum and the CFS serum, and you see that whereas in the controlled serum treated cells, influenza can grow, whereas in the CFS treated serum, the influenza virus cannot grow at all, except for this outlier over here. The rate of growth of influenza virus is completely inhibited. The same for HSP1, which grows nicely in the presence of control serum, whereas in CFS serum, the virus cannot grow. So what we are trying to do here is trying to develop a sort of a biomarker or a diagnostic platform where we can differentiate the ME/CFS patients from the healthy controls or other type of mitochondrial diseases on the basis of their behavior of the serum. So on the basis of these results, we started evaluating what might be the factor in the serum of these patients that is causing this mitochondrial fragmentation. The first thing which came to our mind is reactive oxygen species, but we realized that the reactive oxygen species level is below the critical level what we found in any other person. So reactive oxygen species is not the major source. And the next thing came is interferons. As soon as I say that the cells cannot be infected with the virus, anyone will say that, oh, the cells probably are producing a lot of interferons, and interferon is antiviral, so they are stopping the virus, other viruses to grow. But that's not the case. Here is some data from our collaborator Carmen Seibenbogen's lab. What they did here is that they took healthy control and CFS patients and stimulated the PBMCs with LPS, and then tried to see the different cytokine response, interferon response in the patients, in the PBMCs. And you can see that the interleukin 1, TNF alpha, interferon gamma, and interleukin 5, they all actually, the response of CFS PBMCs are even lower than the healthy controls. So interferon is also not the cause for such a strong antiviral response. The next thing that comes into mind, probably some infection. Yes, of course, mitochondria is the target for many different types of infections. For example, here, if we talk about chlamydia, which is an intracellular obligate pathogen, you see that chlamydia is an ATP parasite. They need tons of ATP from the cell. So what they do is that they fuse the mitochondria. They make elongated form of mitochondria so that they can get tons of ATP from the cell. But on the other hand, if we take herpes virus, which we are very interested in, HHV6, and we reactivate HHV6, for example, over here, you will see that these cells in which the virus is being reactivated by, which is marked by or indicated by the red fluorescent protein over here, you will see that the mitochondria completely changes from a filamentous mitochondria to a punctated mitochondrial form. So on one hand, some of the pathogens, they change the mitochondria to a filamentous structure, to overfilamentous structure. On the other hand, some other pathogens like HHV6, they cause punctated mitochondria. Here is another structured illumination microscopy image to get you a clear idea about how the mitochondria really look like when the virus reactivates. So you can compare the structure of the mitochondria on the left side to the structure of the mitochondria on the right side. Now, we know by now that the HHV6 induced mitochondrial fragmentation is caused by a small non-coding RNA. So you don't need any viral factors, proteins to do that. It's a small 23 nucleotide long non-coding RNA which can actually do the job. So here, for example, these cells are being transfected with either the small non-coding RNA, which is on the top over here, or there are two different variants of the small non-coding RNA, which are only having a couple of sequence differences in the crucial binding region. And you can see that the cells which are being transfected with the small non-coding RNA, they start to fragment mitochondria, and you can quantify this fragmentation. If we completely change the 6 nucleotide sequence over here, we can see that the phenotype of mitochondrial fragmentation is coming back. This we know. Now, many people ask the question, I also ask the same question to myself, that if HHV6 is one of the important virus which is playing a role in ME/CFS, why no one else is trying or finding a clear link between HHV6 infection and ME/CFS? There are many big labs who are trying to understand pathogenic infection in ME/CFS. They are not finding any clear data. And whether there is an active virus infection or there is a latent virus infection which can cause the disease. So to answer these questions, last year we published that the latent virus is present mostly in almost 95 to 98% of the human being, but it's not the latent virus which actually causes the damage. It is the activated form of the virus which causes the damage. So we tried to see if we can find the activated form of the virus in ME/CFS patients by looking into the presence of this small non-coding RNA, which is only expressed or transcribed from the cells when the virus is reactive. And you can see over here that we can show that in a very tiny number of cells, so here the blood cells from ME/CFS patients as well as controls, we take the clots and make paraffin embedded sections, and we try to do in situ hybridization to look for the small non-coding RNA from these viruses. And we can see that 40% of ME/CFS patients show the presence of this small non-coding RNA. And this is telling that there is an active form of the virus in 40% of ME/CFS patients. Now, we heard the last talk from Mike regarding the neuronal link. So we also are trying to see if we can find there is infection in the brain, because these viruses are neurotropic viruses. So we got a nice cohort of samples from Cambridge Brain Bank, where we have almost biopsy sections from entire body of 6 ME/CFS patients and age match 6 matched controls, and we're trying to see if we can find a localized infection of HHV6. And very interestingly, what we have found, it's a preliminary study, but still what we found is that, for example, here there is one case where we have found active viral infection by looking into the small non-coding RNA U14 in the frontal lobe of this patient. We have not finished the study, so we don't know exactly where else also virus is, but at least we can see the frontal lobe contains active viral infection. And there are many literatures in the past which shows that frontal lobe is altered in ME/CFS patients. Here's another case where we see also a HHV6 active infection in the frontal lobe as well as cerebellar dentate region. And here's another case where we find HHV6 active infection in upper cervical cord and the lumbosacral nerve root. So we see HHV6 infection not completely in a form that we can detect it in the blood, but there is localized infection. Unfortunately, our problem is that out of the controls, here is another control which is not really a ME/CFS patients, but this control has Non-Hodgkin's lymphoma, and we see active HHV6 also in the brain stem region. So it will take us time to understand if we can link the active HHV6 infection in these patients or some of the controls to the disease of ME/CFS. But here comes the interesting part, and we also asked the question that we saw in the blood that there are only very few cells which are infected with HHV6. And here also, if we look into the brain or any other part of the CFS patients, we have only very few cells infected with HHV6. But how come we see such a strong, dramatic effect of a lack of energy in ME/CFS patients? This is explained by something called cell danger response, where the cells are very few cells in the body which are suffering from infection or undergoing through some stress conditions can signal the rest of the cells, which are basically healthy cells, to undergo through certain process, which is here a lack of energy. So how we prove it? For example, over here, I come back to the HHP6 reactivation scenario here. You can see here that the cells which are being reactivated are red in color because the red fluorescent protein is coming from the HHP6 backbone. But you can see that the other cell, which are non-reactivating virus during the later stage of reactivation, are also fragmenting mitochondria. Here you can see after a couple of days of reactivation, there is no reactivated cells, the initial viral reactivated cells, because they undergo mitophagy, but the rest of the cells have all fragmented filamentous, punctated mitochondria. You can see another clear picture of this. So what is going on over here? To understand this, what we did is that we took the culture supernatant from the reactivated cells and we put this culture supernatant on healthy cells. So just to be clear here that there is no viral particle formation in the viral reactivation system. So there is no transfer of virus into the target cells. So what we see here is that, again, when we have, so here we have 4 different conditions. In the presence of the virus or in the absence of the virus, we reactivate the virus in the presence of a drug called trichostatin A. So in the absence of the trichostatin A and the presence of the trichostatin A, you see that in the presence of trichostatin A, where the virus is reactivating, we have a strong punctated form of the mitochondria, which you can see that the average mitochondrial surface area falls below. So there is something going on over here that after the viral reactivation, the cells where the virus is reactivating is also undergoing mitochondrial fragmentation. Not only that, the cells which are not having the viral reactivation are also undergoing mitochondrial fragmentation. So we believe that infection, for here, HHV6 is just one example, does something to the mitochondria of the other cells to fragment mitochondria. This may be mitochondrial metabolites. We are trying to understand this process together with Bob Navio from UCSD. We are analyzing the metabolites from the supernatants and trying to understand if similar metabolic changes we can also see in MDCFS patients. We are also focusing on exosomes. We believe that exosomes probably carry certain factors which can cause mitochondrial damage. It can be small non-coding RNAs, it can be proteins. We systematically purifying exosomes and looking into the proteins and the small RNAs. It can be some cellular RNA or pathogenic RNA. HHV6 encoded small RNA is one target. We also have similar sequence similarities with other herpes viruses like EBV. We are looking into that. We have strong belief that antibodies play a very important role. We are looking into not only autoantibodies, but also antibodies probably generated against some of the infectious agents like HHV6 or EBV in certain specific cell types. And these antibodies might target, we have some good data, but I prefer not to talk about it right now. We have also strong evidence that because of the mitochondrial fragmentation, there is a huge change in calcium flux inside the cell, and this calcium flux acts as a signaling pathway to cause mitochondrial alterations. Yeah, that's all of what we are doing in our lab. This is our small group and a lot of different people who are associated with these studies and are supporting with different materials and reagents and funding. Thank you very much for your attention, and I will happy to take questions if you have.